Hi everyone, welcome to Enzymology classes. In today's class, we'll be studying about covalent modification of enzymes. So covalent modification of enzyme is one of the most important type of regulating enzyme activity. So enzymes, like many other proteins, can be covalently modified. So for example, this is an enzyme. So this enzyme can be added with a group by covalent bond. So this bond is covalent. And that covalent bonding with a different molecule, usually a small molecule, converts the protein from a conformationally inactive state to a active state where it can have better activity. So these are some of the examples of covalent modification. Phosphorylation. Phosphorylation happens when a phosphate group is added to an enzyme. For example, glycogen phosphorylase, one of the most common enzymes involved in glucose metabolism. Acetylation. A acetyl group is added to some protein, for example histones, to control its activity. So this acetylation of histone is an important mechanism of gene regulation. Myristylation. So a myristic acid is irreversibly attached to sharp proteins. So these are the membrane bound signaling proteins which are involved in many cell signaling processes. So this SRC or SARC is a myristoylated protein, and myristylation activates the protein. So ADP ribosylation. So ADP ribosylation is ADP ribose group is attached to an enzyme, for example RNA polymerase, to activate it. So farnesylation involves addition of a farnesyl group. So like SARC protein, RAS proteins are held to the membrane by farnesyl group which activates the protein. Gamma carboxylation. So one carboxyl group from bicarbonate ion in the form of bicarbonate ion is transferred to thrombin to activate it. Sulfation, addition of sulfate group, and ubiquitination is one of the most important enzymatic pathway by which a ubiquitin group is added to a protein in order to activate the protein. For example, cyclins are an important class of proteins which are activated by ubiquitination and they control the cell cycle. So out of these different covalent modifications, phosphorylation is one of the most important covalent modification which modifies the proteins. So enzymes, channel proteins and many signaling proteins are activated by phosphorylation, and the phosphorylation of proteins is facilitated by a protein kinase. So a protein kinase transfers a phosphoryl group from ATP to the protein. So similarly, the same phosphoryl group can be removed through the action of a protein phosphatase, which is just a hydrolase, and it removes the phosphate protein. So by the coordinated action of protein kinase and protein phosphatase, proteins can be phosphorylated and dephosphorylated respectively. So over 100 protein kinases is known in yeast and 550 protein kinases are known in human. So they regulate the different cellular processes. The gamma phosphoryl group of ATP is transferred to a specific serine or threonine residue or tyrosine residues on proteins. So protein kinases, they transfer this terminal phosphoryl group from ATP to these residues, serine, threonine and tyrosine, So we remember that these amino acids contain a hydroxyl group on their side chain, and these hydroxyl groups are phosphorylated during with protein kinase. So protein kinases, there are hundreds of protein kinases as we have mentioned before, and they are activated due to some cellular signals. For example, cyclic AMP or cyclic GMP dependent protein kinases are activated by cyclic nucleotide triphosphates. So similarly, phosphorylase kinase is activated by calcium and calmodulin. AMP activated kinase is activated by adenosine monophosphate. Protein kinase C is activated by dialkyl glycerol. Pyruvate dehydrogenase kinase. So a important enzyme involved in regulation of glucose metabolism is activated by increase in ATP, NADH and acetyl coenzyme. So the process phosphorylation and dephosphorylation, they are not the reverse of each other. So phosphorylation happens by kinases, protein kinases. Okay, so this PK stands for protein kinases, which add a phosphate group to a hydroxyl group. That hydroxyl group comes from any of these amino acids. So this phosphate group, this enzyme kinase, they belong to the class of enzyme known as transferases, whereas the removal of this phosphate group is catalyzed by protein phosphatases with addition of water. So they belong to a hydrolase group of amino. So it should not be confused that this phosphorylation and dephosphorylation are the reverse of each other. So they are catalyzed by different enzymes and they are irreversible processes as they are linked to large changes of energy inside the cell. So why this phosphorylation is necessary for the cell? So phosphorylation makes the enzyme activated because of several things. So it adds 2 units of negative charge. So when phosphate is added to an enzyme, it activates the enzyme through electrostatic interaction. It can form electrostatic bond. It can change the structure of nearby amino acids which are charged. Hence, it can activate the protein. So phosphate can form 3 highly directional tetrahedrally arranged hydrogen bond, which are most reactive, and they can just break quickly in response to the different catalytic groups. So they make the enzyme energetic. So this phosphorylation is associated with ATP hydrolysis, and as you know, hydrolysis of a single molecule of ATP, 1 mole of ATP, leads to a release of 12 kilocalorie per mole in cellular condition, and hence this ATP hydrolysis saves 50% of its energy released to the, it provides 50% of its energy to the protein. So the protein becomes energetic, and this energy facilitates a conformational state in the protein which is much more active than the inactive protein. And phosphorylation is a process that evokes the highly amplified effects. So phosphorylation amplifies the signal. So there are a series of proteins which act just downstream to each other to facilitate a big change when a small signal is accepted at the membrane level. So it links the energy status of the cell to the metabolism. So as we know that this phosphorylation of enzymes is linked to ATP hydrolysis. So the cells can link the energy status of the cell to this phosphorylation process. So there are several advantages of this phosphorylation process which happen with this phosphorylation of enzymes. and that's why phosphorylation is an important covalent modification that takes place in regulation of metabolism.